We're now talking about section 5 on the senses for chapter 16 with hearing and equilibrium receptors. Learning objectives for this section. You should be able to describe the structures of the outer, middle, and inner ear. Name the auditory ossicles and explain how they function in hearing. Compare and contrast the bony labyrinth and the membranous labyrinth. Explain the components of the cochlea and how they function in the sense of hearing. Trace the path of a sound wave from the outside of the ear to the stimulation of the vestibulocochlear nerve, CN8. To distinguish between the frequency and intensity of sound. Describe the auditory pathway from stimulation of CN8 to the brain. Describe the structures of the inner ear involved in equilibrium. Explain how the utricle and the saccule detect static equilibrium and linear movements of the head, and explain how the semicircular ducts function to detect rotational movements of the head, and summarize the nerve pathways involved in equilibrium. So the ear structure, of course, the ear detects sound and head movement. Signals are transmitted via the vestibulocochlear nerve, CN8. For the external ear, we have the auricle, which is the funnel-shaped visible part of the ear with elastic cartilage. It protects the ear entryway and directs sound waves inward. The external acoustic auditory meatus, which is the ear canal, extends the tympanic membrane. It's the ceruminous glands produce cerumen, and earwax impedes microorganism growth. The tympanic membrane is the eardrum, and a funnel-shaped epithelial sheet separating the external and middle ear, and it vibrates when sound hits it. And so this picture here, and this will be on your exam for the final here, showing the anatomic regions of the right ear. We have our external ear visible out here, this being the auricle of the ear, then middle ear showing in here. We have our external acoustic meatus right in here. Right through here, and tympanic membrane here, our elastic cartilage. We have our tympanic cavity. We have our oval window, our round window. We have our vestibule, if you will. This goes out to the nasopharynx this way here. We have our cochlea, our shell-like deal looking here, looks like a snail shell. We have the bony labyrinth, this entire space here of the inner ear. Our semicircular canals, our petrous part of the temporal bone, our internal acoustic meatus extended through here. Our facial nerve, CN7. Okay. We have our vestibulocochlear nerve, CN8. That's a key one for us. Cochlear branch and a vestibular branch here. Okay. And of course, the auditory ossicles, the stapes, basically all the way down right in there. And then we have our incus here and our malleus here. The middle ear also will be on your final exam here, showing the little section here that with the temporal bone, petrous part once again here, auditory ossicles from a different angle here, the malleus, incus, and the stapes. Then we have the oval window here, a little closer look than we had before, the stapedius here, the tensor tympani cut here, cut right up here, our round window, our auditory tube, our tympanic cavity, through this region here, and of course the external acoustic meatus and tympanic membrane. Our inner ear, you'll have a section of this on your, actually no, you will not. This will not be on the final, this component here, maybe in your lab though. This showing a little section cut out here for the inner ear. Here we have our bony labyrinth, our semicircular canals, and this little section, this is semicircular canals here with the membranous labyrinth through here, the semicircular duct, the endomorph, our paralymph here is being bone, and then our bony labyrinth, our semicircular canal. Then we have the membranous labyrinth, the semicircular ducts here. Here's our utricle, our vestibule, and our saccule here, our ampullae, all these guys, or ampullae, if you will. Connection to cochlear duct right through here, our membranous labyrinth, our cochlear duct. We have our bony labyrinth, the cochlea, and our cochlear branch of CN8. Our apex of the cochlea contains the heliocotrema. And there's our sectional cut here, showing the membranous labyrinth, the cochlear cut here. This is our spiral ganglion. Here's the bone, a bony labyrinth here for the cochlea, the paralymph, the endolymph, the membranous lymph, the cochlear duct through here, and the spiral organ right down in there. For hearing, you need to know this pathway. This will be on your exam. The pathway from the sound wave to the nerve signal. Sound waves that vibrate via they vibrate the tympanic membrane. Ossicles vibrate and transmit waves to the oval window. Fluid pressure waves in the scala vestibuli push the vestibular membrane, causing pressure waves in the endolymph of the cochlear duct. Specific regions of the basilar membrane move depending on the sound wave frequency. The hair cells are distorted, causing changes in neurotransmitter release. The sensory neurons with the axons in CN8 are then stimulated to fire, and then finally pressure is transmitted to the scala tympani and absorbed by the round window. And so here we have the sound pathways through the ear. So our sound waves directed by the auricle into the external acoustic meatus, causing the tympanic membrane basically to vibrate. And then we have step 2 going here, where the tympanic membrane vibration moves auditory ossicles, the malleus, incus, and stapes, these 3 guys. And the sound waves are amplified. And then 3, right here, the stapes, the oval window, generate pressure waves in the perilymph with the scala vestibuli. And then we get to step 4, where the pressure waves cause the vestibular membrane to move, resulting in the pressure wave formation in the endolymph. Okay? And then basically with the cochlear duct and the displacement of the specific region of the basilar membrane, hair cells in the spiral organ are then, this being the spiral organ here, are then distorted, and that initiates nerve signals basically to the cochlear branch of the vestibulocochlear nerve, which is CN8, and then the remaining pressure basically for step 5 come back through here. Remaining pressure waves are transferred to the perilymph within the scala tympani and are absorbed as the round window bulges slightly, or round window here. For hearing, our cochlear hair cell stimulation, the inner hair cells contain ion channels at their tips, and they tip-link proteins that connect them. The hair cells are bathed in potassium endolymph that is far more positive than the fluid inside the cell. When the basilar membrane moves up, hair cells are pushed into the tectorial membrane. Their tips are tilted. They pulling the tip links. Tip links pull open the ion channel, allowing potassium to diffuse into the hair cell and depolarize it. And the hair cell releases more neurotransmitter from space, excites the sensory neuron, which can fire action potentials, and the basilar membrane then moves down. The process quickly reverses. And here we have our inner hair cells. Basically have our cochlear branch, basilar membrane here, inner hair cell, the tectorial membrane, the endolymph, and of course showing a little more of a better view here. Look in this area here with our tip links, our stereocilia, and then even deeper look here. We have this channel here, right? We have our potassium channel, and we have basically positive potassium on the outside there, the negative on the inside, and higher positivity, of course, for depolarization on the outside. The perception of sound. Sound is a perception of pressure waves established from vibrating objects. This concept of pitch and the frequency and decibels and all that, you will need to know this on your final exam. This whole section, the next couple slides here. Vibration pushes molecules, which transfer the energy from one molecule to the next. Pitch depends on the frequency of the vibrating object. Frequency is the rate of vibration in hertz, and hertz is the cycles per second. Humans can hear from 20 to 20,000 hertz. Below 20, not going to hear it. Really low, like bass. And at 20,000, like treble, really high up there. You're not going to hear that above 20,000, at least generally speaking. Variations in pitch are detectable due to variations in the stiffness of the basilar membrane for the oval window of the cochlear apex. High frequency sounds excite cells in the stiff basilar membrane near the oval window, and the low frequency sounds excite cells in the flexible basilar membrane near the apex. So we have a cool picture showing basically high frequency to low frequency, from the red toward green into blue, and from the base, more for the high frequency, and the apex, more for the lower frequency. This being referred to the relative width of different regions of the basilar membrane. Okay. Now, loudness, which is on your exam, depends on wave amplitude, the degree of molecular compression. You need to know these basic components of loudness. Louder sounds create larger movements of basilar membrane. Larger movements cause faster rate of nerve signals and a larger number of stimulated cells. Temporal lobe's auditory cortex interprets this as loudness. Loudness is measured in decibels, and 0 decibel is threshold for hearing in general, and the energy of sound increases 10 times for every 10 decibel increase. For the auditory pathways, the stimulated hair cells initiate signals in CN8, the vestibulocochlear nerve, fibers ending in the cochlear nucleus of the medulla oblongata, and the secondary neurons transmit signals to inferior colliculus of the midbrain or to the superior olivary nucleus. And then beyond that, the inferior colliculus coordinates head orienting reflexes to sounds. And the superior olive localizes sound and initiates reflexive contraction of middle ear muscles, the tensor tympani and the stapedius. And the superior olive axons also transmit signals to the inferior colliculus. And then for the nerve pathways for hearing, so we get the movement of the basilar membrane, which produces the nerve signals that are propagated, okay, starting here, the cochlear branch here, CN8, to the cochlear nucleus within the medulla oblongata. We're up here to step 2. And 2A, we get some secondary neurons relaying nerve signals, okay, directly to the inferior colliculus, the midbrain, going upward here. And some secondary neurons at 2B here relay nerve signals to the superior olivary nucleus within the pons, which are then relayed to the inferior colliculus in the midbrain. Okay, once again, moving up there. Then number 3 here, we get nerve signals relayed from the inferior colliculus to the thalamus, the medial geniculate nucleus. Okay. And then nerve signals then relayed from the thalamus to the primary auditory cortex of the temporal lobe of the cerebrum for sound perception. Moving along to here. So here we have the medial geniculate nucleus here, our thalamus, of course, here, our primary auditory cortex, our inferior colliculus, our superior olivary nucleus here, cochlear nucleus here. Now, equilibrium, head movement. Equilibrium is just awareness and monitoring of head position. It's monitored by the vestibular apparatus, the utricle, saccule, and the semicircular ducts. Information sent to the brain to help keep our balance. Vision and proprioception, of course, also help. The utricle and the saccule detect static equilibrium and linear acceleration. Semicircular ducts detect angular acceleration. And the static equilibrium and linear acceleration, the macula is a receptor for static equilibrium and linear acceleration located in the utricle and the saccular vestibule. Beyond that, some of the details here, I'm not going to have you worry too much about right now, but the head tilt shifts the autolytic membrane and bends the stereocilia. And when that stereocilia bends toward the canocilium, it depolarizes the hair cells and increases their transmitter release. And that increases that pulse frequency on the vestibular part of CN8, and the opposite reaction occurs if bending is away from the canocilium. It's just a little deeper look at the macula here, the autolytic membrane, otoliths here, our gelatinous layer, and a closer look here at the otoliths themselves. Here's our hair cell, our supporting cells, part of the macula. Here's our vestibular branch of CN8. And then we can see a closer look here at the hair cell, vestibular nerve branches, and the stereocilia and the canocilium. As far as when you see altered head positions, so if you're bent toward the canocilium and if the head goes back, hair cell is depolarizing, increasing the neurotransmitter release, and increased nerve signal frequency along the vestibular branch of CN8 with excitation, the shorter, basically the higher frequency there. Increased frequency. You see a head tilted downward, you're going to see the inhibition, so there's a lot more time between. Hair cells are hyperpolarizing here, inhibiting neurotransmitter release, and there's decreased nerve signal frequency along the vestibular branch of CN8. Angular acceleration, really with angular acceleration for equilibrium head movement. The base of the semicircular canal has a region called the ampulla, and the ampulla contains Crista ampullaris with the hair cells and support cells. The stereocilia and the canocilia, the hair cells, are embedded in this gelatinous cupula. When the head rotates, endolymph pushes against the cupula. The cupula bends the stereocilia and changes the hair cell voltage. If that stereocilia is bent toward the canocilium, the hair cell depolarizes. If it's bent away from the canocilium, the hair cell hyperpolarizes, as we just saw in that image. And the ampulla, you're going to have on your exam a closer look here. This will be on your exam. So in the cupula, your endolymph, your canocilium, your stereocilia, the hair cell, our supporting cells, a Crista ampullaris, and a vestibular branch of our CN8. That will be on your exam. And you can see just a similar look here for seeing if the head is still versus the head is rotating, showing your ampulla here, showing the change and the bending of the stereocilia with the head rotating. And our section of ampulla filled with endolymph here. You see the movement occurring here when the head rotates. Nerve signals are sent to the brain here on that CN8 on the vestibulocochlear nerve. For the equilibrium pathways, signals from the maculae or the Crista ampullaris are conveyed by the vestibular branch of CN8. These axons terminate in the vestibular nuclear cerebellum. And then the vestibular nuclei and the superior medulla help control reflexive eye movements and balance. And the cerebellum helps coordinate balance and muscle tone. And the vestibular nuclei and cerebellum send signals to the thalamus, and the thalamus relays the information to the cerebral cortex for awareness of head position. Here we see our equilibrium pathways. One here starting the vestibular complex to the vestibular branch of CN8, the vestibular cochlear nerve. We're going to go up here to the vestibular nucleus, and then our vestibular spinal tracts, of course, will come down eventually. The skeletal muscles telling them what to do. But here to B as well, we go to the cerebellum, and then we're going to go up here to 3, get to the thalamus, and eventually the cerebral cortex. Then before information is sent out to the skeletal muscles to tell them what to do. So, what did you learn? What is the function of the external acoustic meatus? What are the auditory ossicles? Where are the auditory ossicles located? What's their function? What are the membranous labyrinth structures? What's the specific bony labyrinth structure within where each resides? What are the steps for detecting sounds? Compare the difference in how we perceive pitch versus how we perceive loudness. What are the major brain structures involved in the auditory pathway? What is the function of each? What type of movement do the maculae detect? How do they detect this movement? And what type of movement do the ampullae detect? And how do they detect this movement? And that is the last lecture for Bio 246. Make sure you take a look at the content in course announcements and course assignments to make sure you're fully prepared for your final, and I wish you all the best. Thank you once again for all your time and effort.